I'm Nathalie Delrue from the OECD Secretariat. I'm an administrator of the Test Guidelines Program and of the AOP Development Program. I'd like to welcome you all to this webinar on adverse outcome pathways. There are a lot of participants today, more than 350. Thus, everyone is requested to mute themselves to ensure a good quality of the audio. This webinar is recorded and the recording will be available on our public website. The links will be provided at the end of the webinar. So this webinar is part of a series of webinars that OECD launched earlier this year. We have basically two series of webinars. The OECD webinars on testing and assessment methodologies, which focus on selected ongoing projects, and the webinar today is part of this series. The second series of webinars is the OECD webinars on emerging science. They provide an opportunity to chemical safety professionals to learn and discuss new methodologies developed by research. During this webinar, participants are invited to send questions via the chat box. We'll take a few questions at the end, and we may not have time to discuss all of them, but you are welcome to follow up with the Secretariat with any other questions or need for clarification you may have. The scope of the webinar is to explain what is the AOP framework and why we are developing AOPs. With this webinar, what we would like to do is to encourage collaboration in AOP development, to stimulate crowdsourcing, and to increase the development of AOP networks. This is essential for the success and the usefulness of the program and to ensure mutual benefits. This webinar will be followed up in a few months from now, likely in the second half of 2019, by another webinar on AOPs focusing on their development in practice on the dedicated platform. So this webinar will be more a training on AOP development, while the webinar today is more a webinar on the general concepts, the objectives and the uses of the AOP framework. So let me introduce and warmly thank our speakers today. Kate Willett from Humane Society International will provide an introduction to AOP framework. Jason O'Brien from Environment Canada will focus on the collaborative development of AOPs and will explain how each scientist can contribute to this effort. Dan Villeneuve will describe the various applications of AOPs in chemical risk assessment, such as the use of AOPs to support the development of alternative test methods, or the use of AOPs to support the development of integrated approaches to testing and assessment, also called IATAs. And I will provide a brief overview of the OECD AOP development program before the other speakers go into the heart of it. So AOPs are the central element of a toxicological knowledge framework being built to support chemical risk assessment based on mechanistic reasoning. It is a way to structure the existing scientific knowledge, gather this knowledge in a single platform, and build collective knowledge. So you will see this block structure of an AOP throughout the webinar. An AOP is actually a construct that describes a sequential chain of causally linked events at different levels of biological organization that lead to an adverse outcome. So it can be a health effect or an ecological effect. The OECD AOP Development Program was launched in 2012 overseen by an expert group called the Extended Advisory Group on Molecular Screening and Toxicogenomics, also called EGMOST. This group is made of member country scientists from the regulatory field and academia. It also includes experts from the industry representing business at OECD, as well as NGOs and representatives from the International Council on Animal Protection in OECD programs. Scientific societies can be and are actually represented in this group via their members, but there could be more representatives of this society. Today, the Endocrine Society is the only scientific society formally involved in the group, and we wish to create more opportunities for scientific societies to become key players. The role of EGMOST is to develop an AOP framework, to coordinate the development and the review of AOPs. And you have already noted that the development of AOPs is an important part of the program, but the review of AOPs is also an important aspect that particularly builds confidence in an AOP. However, this review also requires resources, and it is also an area where a broader engagement of the scientific community is also needed and is also key. The EGMOST also provide a platform where AOPs are developed and shared. This is known as the AOP knowledge base, which will be described later on. The role of EGMOST is also to ensure that AOPs inform chemical risk assessment and regulatory decisions. And it provides input to the two OECD groups that oversee EGMOST, the Working Party on Hazard Assessment, and the Working Group of the National Coordinators of the Test Guidelines Program. And lastly, the EGMOST also promotes international participation in the program. Since the start of the program in 2012, the development of AOPs has mainly relied on this group, on EGMOST, but broader engagement and participation of more scientific societies to the AOP development, review, and publication processes is very important, and outreach activities directed towards the scientific communities are expanding. So in order to ensure coordination and consistency among AOP developers, the EGMOST has developed various documents that will help the AOP development and the AOP assessment. These documents include the guidance document for developing and assessing adverse outcome pathways. This document was recently updated, actually in 2017, and the User's Handbook supplement to the guidance document. This document also was recently updated last year. And these documents are available on the OECD public website. So what's the AOP development process? The first step is to look at the guidances or to follow a training on AOPs. There are ad hoc training sessions organized by a subgroup of EGMOST, and you can also find an online training which is available on the internet. Knowledge is then assembled, evidence is weighed, and uncertainty described. And when fully developed, the AOP is scientifically reviewed. That's the last step of the development of an AOP. So the AOP knowledge base is the IT tool supporting the development of AOPs. The main entry point for the AOP knowledge base and its different modules is the AOP portal here. This AOP portal is a search engine for all AOPs, and it allows browsing of review reports too, which are available there in this portal. So as you see, there are 5 modules in addition to the portal, and I will focus more on the 2 main modules, the AOP Wiki and Effectopedia. Effectopedia is a module that captures quantitative information and provides standard visual representation of AOPs and associated test methods. The AOP Wiki is the default go-to module for all qualitative AOPs. Most AOPs are developed on this platform, which is also the entry-level module for evaluating and reviewing AOP scientific evidence. So anyone can access and read through the wiki at aopwiki.org. If you would like to start commenting back to AOP authors who have developed AOPs in the wiki, you can simply register on the wiki platform so that the wiki team can keep track of who is commenting, and you can enter your comments. If you would like to begin entering your own AOPs, your own key events and key event relationships, then you will need to ask for a right access through the AOP Wiki website or through the Society for Advancement of AOPs.
Thank you very much. And thank you all for being here, and thank you, Nathalie, for the introduction. So my presentation is going to cover the basics, the why, what, and a little bit about the how of AOPs. I'm going to introduce AOPs as the basis for predictive toxicology. I'm going to introduce the AOP concept, the essential elements of AOPs, and a very brief on building and using AOPs. And then I'll go over just very briefly the available guidance, tutorials, and classes that can help you build and evaluate AOPs. Okay, first, why do we need AOPs? Well, what we do need is there's a lot of forces that make us realize that we need to transform the historical way of assessing chemical safety for a number of reasons, including a growing concern over the lack of information about tens of thousands of chemicals that are in the environment, and concern about the high failure rate of drugs in spite of increased spending on research and development, and the need for faster, more relevant approaches across chemical sectors. Therefore, to increase the efficiency and improve safety decisions, there's a need to move from a system of empirically measured adverse outcomes to a predictive system based on the measurement of upstream biological events, coupled with an explicit biological linkage to potential adverse effects. At the same time, there's also a need to better leverage our existing knowledge. We have so much data. Biologists have been generating an increasing amount of data for decades. In fact, a 2014 Nature blog estimated that global scientific output doubles every 9 years. There's way too much information for us to digest, let alone comprehend. And where is this data? It's largely in journal articles and reports, or it's in laboratory notebooks, agency archives, or it's stored in institutional or government databases. It follows that if we had better access and better organization of data, it would lead to better understanding of biology and a better approach in terms of how to use the biology. If we had a better understanding of what we know and what we don't know, be able to design better research and testing approaches. But how do we get from here, where the data is largely in PDF format, it's fragmented, siloed, and with a good portion of it considered proprietary, to here, Where the data is accessible by searching, it's machine readable and linked together in a comprehensive way. If we did this, it would facilitate collaboration, it would avoid duplication of research and testing, it would help us guide test strategy design, it would support model building, and it would also help us guide and focus future research. This is a problem that AOPs are designed to help solve. The idea of the AOP framework began as a practical solution for a practical problem. How do you use molecular information to make better decisions about chemical safety to protect humans and the environment? However, it turns out that the AOP framework is actually much more than that. It allows us to more fully utilize existing knowledge, capitalize on advances in chemistry, biology, and engineering to not only design better approaches for assessing chemical safety, but also to better focus research, understand disease, and improve drug discovery. The AOP framework is a way of linking changes at the molecular and cellular levels to downstream consequences and potential adverse outcomes. An AOP documents the changes that follow after an experience of a stressor by a cell or tissue. Through a series of key biological steps or processes that may occur at the tissue, organism, or even the population level. And finally, to the possibility of an adverse result or outcome. Another way of thinking about this is that the AOP is really a map of the sequence of events that happen after a perturbation. An AOP begins with the initial interactions of a stressor with a biomolecule in a target cell or tissue, the molecular initiating event, progressing through a dependent series of essential and measurable intermediate events or key events. The key events are linked together through a description of the key event relationships and culminating with possible adverse outcomes, which can be at different levels of biological organization, outcomes that regulators generally use to make safety decisions at the organ or individual level for human health decisions, and at the individual or population level for ecological health decisions. Another way of thinking about AOPs is as a framework for organizing data. All types of information that pertain to a given biological event or process at all different levels of biological complexity, from the quantitative structure activity relationships or structure activity relationships to molecular data like gene or protein expression, any kind of omics data, to more complex cell and tissue-based tests, to whole organism observations like typical guideline studies or clinical data, or even to population-level data like epidemiological studies or ecological studies. In this way, all these types of disparate forms of data can be related to each other and turned into knowledge about a particular biological process. The ultimate goal of this framework is to be able to link the upstream molecular information to the downstream effects well enough that the latter can be accurately predicted from the former. So say you'd like to start building an AOP, which would be great if you would. How do you begin? Well, you can start anywhere. You can start at the molecular initiating event, you can start at the adverse outcome, or you can start in the middle and build towards one of these anchors. Where you start usually depends on your field of expertise, but the basic process is the same regardless. A pragmatic decision has been made to define an AOP as one molecular initiating event leading to one adverse outcome. This is just an artificial convention, a simplification of biology, if you will, to give you a place to start. Okay, so how do you start? You begin by gathering all existing information, not every detail, but the critical steps or checkpoints in the biology. And because an AOP covers a wide range of biological complexity, it's unlikely that one person is going to have expertise in all of these areas or in all of this biology, and therefore collaboration is encouraged. And the AOP framework and the wiki and the documentation is all designed to encourage collaboration. The next step is to document and capture this information as elements of an AOP in the AOP wiki. As part of this process, you evaluate the information that supports your key event relationships. And this is done using a weight of evidence framework that has been modified to adapted to be specific for AOP evaluation. And there's extensive OECD guidance about how to do this. And finally, when you are ready, and if you want to, you can enter the OECD evaluation process, which includes a rigorous scientific review of the information underlying the AOP. As you're developing your AOPs, it's important to keep in mind that there are some fundamental principles of AOP development. One is that AOPs are not chemical specific. Chemical specific information may be used to inform the AOP, but the AOP itself is the biological pathway and is independent of the stressors that can affect it. Another concept is that AOPs are modular. Key event and key event relationships are designed so they can be shared by multiple AOPs. As a pragmatic construct, an individual AOP is composed of a single sequence of key events and key event relationships leading to a single adverse outcome. As I mentioned before, this is just a pragmatic decision to simplify the process. However, the fourth principle, that AOP networks will emerge and are the basis for prediction, is extremely important. Networks are likely to be the functional unit of prediction for most real-world scenarios. I'll talk about that a little more in the next slide. And finally, AOPs are living documents. They are never finished, and AOP descriptions are expected to evolve over time as we learn more about the biology. Okay, this slide exemplifies how networks emerge as more and more AOPs are developed. This diagram shows three related AOPs: ER antagonism, aromatase inhibition, and AR agonism, androgen receptor agonism. All three of these AOPs start out with unique molecular initiating events, and the upstream key event and key relationships are unique. However, at a certain point, all three of these pathways, they merge, and they intervene with the same key event here, which is hepatocyte-reduced vitellogenin production, and after that, they follow the same pathway, so they share key event relationships and key events between this key event and the adverse outcome. And in this way, when you share a key event or key event relationships, networks begin to emerge. And as networks emerge, the AOP system begins to resemble biology. Okay, so regardless of their state of development, AOPs can be useful for a number of things, including, as I've shown, organizing and relating data and different types of information. They can be useful in generating hypotheses for answering particular questions. They can be very important for focusing research, because as you develop AOPs, you not only learn what you know, but you learn what you don't know, and this can help point you in the right direction for future research. They can help in the identification of optimal key events for assay and integrated test strategy design to answer particular questions. They can be very helpful in supporting weight of evidence interpretations and decisions because they provide a transparent documentation of the information underlying these decisions. And furthermore, as quantitative information is accumulated in AOPs, they can aid in design of predictive models. As Nathalie mentioned, central to the OECD AOP framework is the AOP knowledge base, an information hub that's designed to collect, organize, and evaluate AOP information. The knowledge base consists of several software packages, the most developed of which is the AOP Wiki, and most of the guidance and materials are concerned with the AOP Wiki, and this is where you would most likely begin your interaction with the knowledge base. The AOP Wiki is a database for capturing and organizing all information and supporting documentation for key events and key event relationships. As the name indicates, the AOP Wiki is wiki-based, and like Wikipedia, is meant to be crowdsourced and developed by as many people as possible around the world. The wiki is supported by extensive documentation, tutorials, and an online course. It's designed to enable rigorous evaluation and scientific review of the biological information. It's been publicly available since 2014. Okay, now I'm just going to briefly go over some of this tutorials. These slides will be available after the webinar, as Nathalie mentioned, as a reference, so I'm not going to go over these extensively. The most important guidance for developing AOPs is the OECD User's Handbook. And this is a supplement to the wiki that contains instructions about how to use the wiki and also some support in evaluating key event relationship, the weight of evidence evaluation for key event relationships. Here also listed are a lot of websites that contain a lot of information about AOPs. And there's also a number of training videos and tutorials available online through these links. And finally, there is an online course available, and the course will actually walk you through how to use the wiki to build an AOP, and also the steps involved in reviewing key event relationships. The course has 3 modules. It has an introduction and overview. It has a tutorial that has internal quizzes to help you to help you see how you've comprehended stuff. And then finally, at the end, there's a self-exam if you want to test how well you've understood the material. And this, you can download it from the Human Toxicology Project website, or you can run it directly from the AOP Wiki. Okay, a brief summary here. I hope I have convinced you that the AOP framework can be quite useful for a number of things. In summary, the AOP framework is a formal process to collect, organize, link, and evaluate biological information. And it began as a practical solution to a practical problem: how to use mechanistic biological information to support better decisions about chemical safety. But it's really a lot more than that. It's the basis for predictive toxicology. It allows the use of upstream molecular information to predict the likelihood of downstream events. It's a transparent, highly curated living document that represents our current biological knowledge globally. And it's also useful in focusing and directing research, including disease research. So the AOP framework, the utility of the AOP framework is dependent on wide adoption. It can be a lot of work, and it requires a wide range of expertise. So collaboration is essential. And the AOP Wiki and framework are designed to facilitate this collaboration. The ability to truly capitalize on collective biological knowledge will transform not only chemical safety assessment, but also medical research, and therefore the payoff is immense. In order to make all this work, the AOP framework needs your participation. So I really hope you will consider entering what you know into the AOP Wiki and help make this a much better platform for all of us. All right, thank you.
Thank you very much, Kate, for this presentation. Now we will move on to the presentation from Jason O'Brien.
Hello, I'm just going to grab the screen here.
Hello, Jason.
Can you see my screen?
Yes, we can. Thank you.
All right. So thanks very much, Kate, for that introduction to the AOP framework. I'm going to be reiterating a lot of what Kate has said about all the benefits of the AOP framework, but I'm going to be focusing specifically on the collaborative aspects of the AOP framework. So much of what what I'm going to say is going to be repetitive with what Kate was saying, but with a little bit more detail. So, as Kate mentioned, the AOP framework was designed with collaboration in mind. There's several aspects to AOP collaboration. One of the main things when we first started was to be able to create a framework where people can work together to develop the AOPs. But as Kate mentioned, it's become much more than that. It's also a place where we can share knowledge and also collaboratively guide our research together. And this is all done by working together. So the AOP framework in general runs all the way from molecular initiating event all the way up to the organism or population level of biological organization. And it's very rare for a single person to have expertise in all of these areas of biology. So often AOPs require collaboration between large groups or teams. These teams can consist of molecular biologists, cellular biologists, physiologists, risk assessors, ecologists, epidemiologists, and so on. So it's very rare for a single person, though it can be done, but usually requires groups. Now there's many different types of groups within your own research institution or within a university, but we're really hoping to get participation from scientific societies into the development and application of AOPs, because societies are really well suited for collaborative development of AOPs. Societies are large groups, and they usually have the expertise across all of the range of biological organization that is required for the development of AOP, and they have the most cutting-edge knowledge in these areas. And one of the things that we're really trying to point out is that the benefit is not just to the regulatory community. There's a lot of other benefits for your participation in the AOP process. So, of course, the major goal of this is to support the uptake of new approach methodologies, such as in vitro and high throughput methods for the purpose of risk assessment. But there's many other benefits, such as just the ability to organize your science into this framework really helps you understand the state of the science and understand where you can contribute the best and where more work is needed. Not only that, it helps you communicate the science better. It's a much more easily understandable framework than just a collection of scientific journals, even compared to a review article. As I mentioned, when you organize all the information this way, you can really identify knowledge gaps, and this helps you direct where you can focus your research efforts. And all of this will help you to generate new knowledge in your field of research and to help really push the science forward. So the first aspect of collaboration I want to talk about is collaborative development. And as Kate mentioned, the most common way to develop AOPs is through the AOP Wiki. Now, almost all aspects of AOP development can be done through the Wiki. And as we've mentioned, the AOPs were designed in a modular way so that the AOPs themselves can be broken down into smaller, more manageable components, such as the key events themselves or the key event relationships. And this allows the groups to divide the work between each other. And you can also, every single AOP is broken down into separate key event pages and key event relationship pages. And this allows you to assign the work to different members of a group, and you can all work on this in real time together so that you can efficiently collaborate to bring a full AOP together. Now, in order to use the wiki, you need to have access to the wiki, and there's different access requirements. The wiki itself and all the information in the knowledge base is freely available worldwide. You don't need any user registration. All you got to do is go to aopkb.org or aopwiki.org, and you can view everything, all the AOPs, the key events, the key event relationships, and all the documenting and supporting information.
Eu não meti a password.
And then if you want commentator access, that means you can comment on any of the AOPs that are being developed. You just need to register an account and verify your email address. This is a very simple process. It can usually takes less than a day. And this will allow you to contribute comments to all of the AOPs that are being developed. But if you want to actually create AOPs, create key events and key event relationships, of the various components of AOPs, you need to get author access. And there's two main ways you can do this. One is through the OECD. You can submit an AOP work plan. This process takes a little bit longer, but there's several benefits. You get the support of the OECD EGMIS group to review your work plan and review your AOP and guide you through the development process. And another option is getting author access through the Society for the Advancement of AOPs, the SOAP. This is a faster process, but you don't quite get the same support. All of this information, there's more details of this actually on the wiki itself. So if you're interested in getting these different levels of access, please take a look at the wiki and find out. Once you do get your access, there's many different ways that you can contribute to the AOP knowledge base. Of course, you can create full AOPs, but sometimes if you don't have all the expertise required, you can just contribute parts of AOPs or even single components. Another aspect that we haven't talked about much is borrowing and sharing all of the different components of AOPs. And you can also use all the information in the wiki to help guide your research so that you can generate data that can support some of these AOPs. And all of these activities will help share and even generate new knowledge, as I'll show you shortly. So, as I mentioned, making a full AOP often requires the effort of a full team of experts, since it covers many spans of biological organization. However, if you feel like if your team or yourself doesn't have the required expertise, another option is to create partial AOPs. You could create, for example, if your area of expertise is in the molecular area, you can create the first half, and then hopefully you can coordinate with another group of authors, or you could just hope that another group of authors will kind of pick up the ball and complete the end part of the thing. But eventually, because of the modular nature of the AOPs, once these two components are built, they actually combine together to form a full AOP. Now, you can do partial AOPs, but really you can break down to even smaller units. Of course, you can do individual key events themselves, but key events themselves are really just descriptions of how an event is measured. They're useful, but the most useful unit that you can contribute is the key event relationship. Now, this information is going to be covered in later webinars where we really get into the details of how to make key events and key event relationships. But for now, it's helpful to know that the key event relationship contains the experimental evidence that links the two key events together. It contains causal evidence, and it also contains a weight of evidence evaluation, so we know we can assess the quality of this evidence. And so the key event relationship really is the principal unit of extrapolation. It's how you move from one, or the evidence that proves or supports the movement of upstream events to the downstream events. So if you really want to contribute to the AOP knowledge base and feel like you have limited resources, this is one of the most useful contributions that you can make. So if a whole bunch of different authors all create single key event relationships, similar in the way of the partial AOPs, if there's overlap in them, they can combine together to form a full AOP. Now, similarly, there's no sense in recreating what has already been made. If you want to make an AOP, and someone else has already made part of it, Kate showed an example of how some of them converge towards the end. But in this particular example, let's say there's some upstream key events that have already been described that would fit an AOP that you'd like to make. Well, you can simply just borrow those key events and make them part of your AOP. And this is where the AOP networks really start to form. When you start borrowing and sharing key events and key event relationships, this is when you really start to generate new and novel and useful knowledge. So in this particular hypothetical example here, we have 5 separate AOPs, but as you can see, some of them have some overlapping components. They have some shared key events and some shared key event relationships. Now, on their own, they form 5 separate AOPs, but when you combine them together into a network, 3 new paths actually emerge from these AOPs, just from the fact that they share components. And it's in this way that AOPs can actually form new knowledge that didn't exist before. We conducted a recent study on the state of the whole AOP knowledge base about a year ago in April. And at that time, there were 187 separate user-defined AOPs. And that's what this figure over here demonstrates. When you combine them together, they form a large network. And even though there was only 187 separate AOPs, these actually formed 9,000 unique MIE to AO paths. So anytime you contribute any sort of knowledge to the AOP framework, you're not only sharing this useful knowledge with your peers and the risk assessment community, you could also be contributing to the generation of new knowledge that might not have existed before. So a couple examples of AOPs that have been built in these types of ways. Now, many of the AOPs, as we've mentioned, have been built by groups of people. And so most of the AOPs in the knowledge base are built this way. But a really, really good example is this Parkinsonian motor deficits AOP. There's a publication that goes along with it, and this AOP was 7 key events and 8 key event relationships, and this was a collaboration between several different organizations. And if you look closely at the author list, you'll see that one of the authors was actually a whole working group, the European Food Safety Authority working group. And so there was a collaborative effort in building all of the separate components of this. And this is probably the most common way that AOPs are made. Another example, as I talked about before, is partial AOPs. The Genetic Toxicology Technical Committee for HESI are experts in genotoxicity, but they don't feel they have the expertise in the population level. So they've created partial AOPs that lead from molecular initiating event up to what they call the genotoxic adverse outcome. And so here are a few examples. They've made one for tubulin binders, topoisomerase poisons, and topoisomerase inhibitors. And so they're developing these partial AOPs that go up to the genotox endpoint, and it is hoped that someone else that is more of an expert in the population level will pick up that endpoint and bring it all the way to the final adverse outcome. And here's another excellent example of sharing key events and collaborative development. Here are 3 AOPs, and they at first seem somewhat unrelated. Well, 2 of them are close: liver fibrosis, lung fibrosis, and impaired learning and memory are the adverse outcomes for these three AOPs. And these three AOPs, among others, we found that many different AOPs were using various descriptions of inflammation. And in fact, inflammation was one of the most common key events being used in the AOP knowledge base, but everyone was using a different version of inflammation. So when this was noticed, a workshop was held for the development of a common description of inflammation. And this is the results of this workshop are published in this article right here, so I recommend you take a look at that. But the outcome was that they developed a universal definition of inflammation using three key events, these three key events right here. And what that allowed is this more comprehensive network of AOPs that described three AOPs in three different tissues, all using the same description of inflammation. So this is an excellent example of collaborative key event and key event relationship development, and also an excellent example of key event sharing. So those were some examples of AOP development. But as I mentioned, development is not the only benefit or the collaborative aspect of AOPs. One of the really important ones is organizing your information, your science knowledge. In fact, that's one of the primary purposes of the whole AOP framework, is to efficiently and effectively organize complex experimental evidence. So an excellent example is if you go in the literature and try to find information on endocrine disruptors and all the pathways that are affected by endocrine disrupting chemicals, you'll find hundreds, if not thousands, of articles. And how do you organize these? Even review articles are pretty unwieldy sometimes to truly grasp and organize all of that information. And so that's one of the major goals of the AOP framework, is to take all of this evidence and organize it into a framework that's easy to interpret, easy to share. It's machine readable. And so here is basically a summary of this endocrine disruption network of AOPs. And each of these boxes and arrows represents at least one journal article, if not more. And in addition to directing you to where that information is, it also, we haven't talked about it much, but there's also an assessment of the quality of that information in each of these arrows as well too. So not only can you find out what information is available, how it's connected to each other, you can also learn about what is the quality of that information. And this allows you to really, really share your information. It makes it much more accessible to non-experts. So this was one of the primary goals for putting the information in this format, so we can share it with risk assessors who are not necessarily experts in the molecular realm. This makes the causal relationships between all the different key events very clear. And as I mentioned, there's a weight of evidence is communicated. The quality of the information is also communicated. Now, once you have this information, it's great for sharing, it's great for communicating, but it's also really good for directing your own research and working on large research projects within a community. As I've mentioned already, it's a really great way to synthesize the current knowledge. It has a weight. Each of the relationships has a weight of assessment associated to it. So here you can see that several of these relationships have very strong weight of evidence, but you can also identify knowledge gaps. So in this particular example, there's not very much strong evidence linking these two key events. So this allows you to identify areas where you can focus your research so that there's no duplication in efforts. You're not doing what people have already done. You're not adding information to an area that's already well understood. You can really identify the weak points in the area and generate data that has the greatest impact. And in addition to identifying these data gaps, and again, we're not going to go into too much information about this in this particular webinar, but there's the AOP framework and the guidance documentation can help suggest what types of experiments can be used to strengthen these weak gaps. There's different types of causal evidence that are particularly useful for supporting AOPs. For example, and I'm not going to get into too much detail about this, but there's types of evidence called biological plausibility, or dose, incidence, and temporal relationships, and evidence that supports the quantitative understanding of the key event relationships. So in summary, the AOP framework is not only is it good for organizing the information for linking molecular to adverse outcomes, it's a great collaborative community that has many advantages to using. So there's, of course, the collaborative development of AOPs. You can create full AOPs, partial AOPs, even single modules of key event relationships you can borrow and share, and all of this helps generate new data in the knowledge base. And then once you've done this, it's an excellent tool for sharing knowledge in a very easy to understand way. It makes this information much more accessible to non-experts. All the relationships and the quality of the evidence supporting those relationships becomes much more clear. And you can also use this information to help guide your own research. It gives you the most current synthesis of the current knowledge. Again, the weight of evidence is assessed. You identify knowledge gaps and identify areas where your research can have the greatest impact. And that's all I have for AOP collaboration.
Many thanks, Jason, for this really nice presentation. We will now move on to the presentation by Dan Villeneuve.
All right, hello, can you hear me?
Hello, yes, very well, and we can see your screen, Dan. Thank you.
Great. Okay, so in the third presentation, what I wanted to focus on is the application of adverse outcome pathways in chemical risk assessment. And so thus far, just to recap what you've heard so far, we know that adverse outcome pathways are a conceptual framework to help us organize existing knowledge and better utilize that knowledge in various types of regulatory decision making to guide research and so on. You heard about 5 principles of adverse outcome pathway development, notably that AOPs are not chemical specific, that they're developed in a modular fashion with sort of linear AOPs from molecular initiating event to adverse outcome as a pragmatic unit of development and review, but all of that really building toward networks that capture our more complex understanding of biology and interactions that occur there. And that finally, these are living documents, and that we continue to add new knowledge, new support, new evidence to these as they develop. Then you also saw in Jason's presentation that this is a modular, or that this framework and the knowledge base employ a modular structure that's designed to support collaboration and crowdsourcing. And so we focused a lot so far on content developers, how they can contribute to the knowledge base. What I wanted to do in this last presentation is talk a little bit about the content consumer. How is it that AOPs are being used currently, and what are some of the potential advantages and ways that they're being employed? And so AOPs themselves, as a knowledge integration approach, are very flexible tools. They can be brought to bear on a lot of different sorts of questions, address different applications. I'm not going to go through every possible application today. I'm just going to touch on some of the more prominent ones, give you some examples of how they've been used in developing alternative testing approaches, supporting more hypothesis-driven testing strategies as frameworks for organizing evidence, uses in environmental monitoring, and then talk about some of the risk-based applications of AOPs where a quantitative understanding is sufficient. So first, in terms of alternative testing approaches, one example that Jason showed earlier was a series of AOPs related to disruption of thyroid hormone signaling. And so that series of AOPs was actually used in the process of trying to develop alternatives to whole animal test, the amphibian metamorphosis assay. This assay was developed to screen for the ability of chemicals to impair the synthesis or metabolism regulation of thyroid hormone. Of course, the animal test costs in excess of $75,000, uses in the neighborhood of 300 plus tadpoles, and takes 3 weeks or more to perform, let alone to analyze the data. So there was certainly interest in coming up with higher throughput replacements for that test. In order to do that, one of the things we needed to understand if we were going to replace this whole organism with a battery of, say, in vitro screening assays is how many different ways chemicals can interact with the biological system to reduce the production of serum thyroid hormones or to impair things like deiodinase that are needed to activate the thyroid hormone in target tissues. And so investigators that were working on this problem basically laid out a series of adverse outcome pathways. They identified various molecular initiating events, ways that chemicals could perturb this biological system. They identified the serum thyroid hormone concentrations as a key hub in this network of adverse outcome pathways. And they also mapped those outcomes of those perturbations to effects in a number of different species. So we had used the amphibian as a model organism that we knew metamorphosis is dependent on thyroid hormone signaling, but we also know that impaired thyroid hormone production can lead to cognitive impairments in rodents and other types of mammals, and also that plays a key role in fish, for example, in development of the swim bladder and and auditory sensing, and so plays a critical role in young of year survival in fish. So we both have sort of a list of the types of perturbations that can result in changes in thyroid hormone signaling. We know adverse outcomes in a wide variety of species. And so this information was then to use this or to support the development of a series of screening assays that probe chemicals for their ability to interact with or perturb these various molecular initiating events, and therefore indicating that they have possibility or some likelihood to potentially cause these types of adverse outcomes in different taxa. So basically, again, supporting the use of these as alternatives to that whole organism test. Sort of building on that same theme, in trying to replace the assays that were used for skin sensitization, an AOP-based approach was used. So there was pretty good consensus that no single non-animal method could replace animal testing for skin sensitization. And so instead, a combination of different assays that represented sort of different key events along this adverse outcome pathway, all the way from direct interaction with peptides and reactivity with those peptides that can be screened in a simple in vitro assay, to activation of dendritic cells or keratinocytes, and then furthermore looking at T cell proliferation. And so in this frame, these alternative assays were placed into an adverse outcome pathway context and then used in an integrative fashion to pull together the weight of evidence that supports the idea that this chemical may act as a skin sensitizing agent. And so that's one of the ways that AOPs are being used to support so-called integrated approaches to testing and assessment, or tiered testing strategies. So AOPs in this context really help to integrate existing information and put it into context. Because the key events in the AOP are actually observable, we can employ them to actually formulate testable hypotheses. We can predict what we should see and actually measure that. And again, as mentioned, using these AOP-based approaches can help target the data collection on the areas that we may be missing data, or allow us to collect additional tiers of information that help confirm the hypotheses that we are generating based on the AOP. Just as another example of this, there's another AOP in the Adverse Outcome Pathway Wiki that describes the linkage between cyclooxygenase inhibition and reproductive failure in birds due to eggshell thinning. In this case, we can use something like a high-throughput screening battery that includes an assay for cyclooxygenase activity. where we see chemicals with that activity, we may have reason to suspect, based on our AOP, that they could cause reproductive impairment in birds. But do we want to jump right into doing an avian reproduction study at a cost of $250,000 and 30 weeks to perform? Instead, we might use the AOP to guide us to some intermediate endpoints that we could look at, say in vivo in a 96-hour assay looking at prostaglandin concentrations to give us an indication of whether exposure to this chemical in vivo may actually produce the biological changes that we would expect to progress along this line. And so we can use this in a tiered approach to help gather more evidence before running this very expensive long-term test if we need it at all. So those are some examples of how AOPs have been used for alternative assay development and integrated approaches to testing and assessment. Another way that AOPs have been employed already in various regulatory decision-making relates to just organizing and evaluating evidence for causality. There are two examples of this where this has actually been put into practice. One is in looking at the role of particulate matter in the induction of metabolic syndromes like diabetes. And a second, looking at the linkage between neonicotinoid exposure and adverse effects on pollinators. These are both high visibility topics of concern to the regulatory community. They're sort of either health or ecological sort of epidemiology studies where we've got some observation of adverse effects occurring in a population. We've got some potential exposures that are associated with those, but can we actually develop the evidence that supports a causal relationship between those? And so in the process of doing an integrated science assessment where we search the literature, select studies, and evaluate the study quality, and try to come to some scientific conclusion regarding the causal relationship between stressor and particular outcome, the AOP provides a framework to aid in the evaluation, synthesis, and integration of the evidence. So, for example, in the case of connecting particulate matter to these metabolic syndromes, we have a wide range of different data types, everything from sort of in vitro studies to controlled and uncontrolled trials, experimental work, observational studies like epidemiological evidence from humans, as well as controlled human exposures, animal toxicology. And so a wide variety of different data and information that sort of gives you different pieces of the overall picture. And so an AOP framework here was used to help identify how these different lines of evidence fit together and whether they actually supported this causal linkage between particulate matter exposure and these outcomes. Likewise, in the case of pollinators and their exposure to neonicotinoid insecticides, again, a very complex literature, everything from very detailed sort of mechanistic studies at the molecular level, how these pesticides interact with the nicotinic acetylcholine receptor, how that affects downstream signaling pathways, and so on. But then also studies up at the level of entire colonies and death or failure of the colony on behaviors like foraging and learning behaviors. And so how do all these different lines of evidence relate together? How do they support one another? Where are the gaps in our knowledge? And so this type of information was organized into an adverse outcome pathway network that also integrated potential non-chemical stressors that could interact with these various key events, so things like varroa infestation or weather events. And so this all contributed to an assessment of potential contributions of neonicotinoids to impacts on pollinators. Another area where the AOP framework has been employed productively is in the area of bioactivity-based environmental monitoring. So if we go out to collect a water sample or a sediment or an air sample, we can extract chemicals from that. Some of those chemicals we can quantify using analytical methods, but other chemicals we may not have standards for, we may not have the proper methods for, or we may not understand exactly how these chemicals are going to interact in a mixture. And so we can take that extract, test it directly in various bioassays, such as high-throughput assays, and then we can use the adverse outcome pathway framework to associate these molecular level perturbations, these cellular perturbations, with potential hazard. And so an example of that was conducted on a site on the South Platte River. This is a site downstream of a wastewater treatment plant discharge. A water sample was collected, an unknown chemical mixture extracted from that, and that sample was then tested in the Adagene battery of ToxCast assays. So this includes about 80 or so different molecular level perturbations or pathway-based responses to that chemical. We could then look at this profile in terms of the chemical or the biological activities that were associated with that sample, and there were basically 6 molecular initiating events that were identified as being impacted by this complex mixture. We took those 6 molecular initiating events, identified where they occurred in the adverse outcome pathway network that's represented in the AOP wiki. We then extracted out all the AOPs that linked to those 6 molecular initiating events, filtered that network to select those AOPs that we felt were relevant to aquatic organisms, and based on that, we're able to identify embryo lethality and potential reproductive failure as hazards that might be associated with exposure to that mixture. And we can also use these intermediate key events to identify some markers or indicators that we could use to look at fish or other organisms out in the field to get an understanding of whether some of these predicted hazards, whether we see evidence that exposure to these chemicals is actually leading us down one of these pathways that could result in these adverse outcomes. The final example of how AOPs are being used relates to moving beyond just identifying a hazard, but actually trying to understand whether the concentration of the stressor we're exposed to is actually sufficient to push that perturbation all the way to the end of the pathway and cause an adverse outcome. So again, a lot of our AOPs are described qualitatively with the assumption that we're going to drive everything to the end, and so it's really, it's a hazard assessment. It's sort of like standing on a cliff. Certainly there's a hazard there, but risk is determined by things like how hard you're being pushed or how hard the wind is blowing, whether the individual's balance is good enough, whether there's a guardrail, all these types of things. And so we want to understand for AOPs how exposure interacts with that biological system, what factors may confer greater or lesser vulnerability to those stressors, and then how to assess whether we're at that tipping point or whether we're not going to get to that adverse outcome. And so in describing these key event relationships, and again, this is an example of an AOP that's in the AOP wiki. This one links inhibition of the enzyme aromatase to reproductive failure in oviparous vertebrates. In this case, in describing this key event relationship between the molecular initiating event and one of the intermediate key events, concentrations of the steroid 17-beta-estradiol in plasma, we actually have defined the quantitative relationship between these two key events. So for a percent inhibition of this enzyme aromatase as measured in an in vitro assay, we can actually predict, based on this mathematical relationship, the likely concentration in the plasma. we walk down the pathway, there are additional key event relationships with similar types of quantitative understanding. Here, just skipping to the third series in the pathway, linking production of this egg yolk precursor protein vitellogenin, concentrations measured in blood, to average fecundity in the fish. Again, we can use this mathematical relationship to take and extrapolate along this pathway for a given piece of information. And so for AOPs with this kind of quantitative understanding, that allows us to, for a novel chemical, bridge all the way from identifying the concentration of that chemical in an in vitro assay that produces the same level of response that we see for a reference chemical, in this case the aromatase inhibitor fadrazole. Based on that percent aromatase inhibition, we can then extrapolate all along this pathway to a prediction of impacts on cumulative fecundity or even out to the population level. And so, while there's certainly some uncertainty in this estimate, it gives us a first-tier sort of look at what we would expect as a benchmark dose that we could use for a first-tier sort of risk assessment. So overall, again, these were just a couple examples of how adverse outcome pathways have been applied to date. There's lots of different ways that people can use these, and through interactions at meetings and so on. We're amazed, I think, at the variety of different applications that people are finding for using this kind of organized knowledge. And so AOPs truly are a multi-purpose tool. They've already been applied in many different contexts to support chemical risk assessment. And overall, the greatest current limitation to their application is really just the limited number of high-quality AOPs that are currently described and available in the Adverse Outcome Pathway Wiki. And so the goal of this webinar series and many of our other activities associated with the AOP program is to really remedy that, to increase the amount of information that's been synthesized and organized according to this framework, and make that information accessible to those that want to use it. And so certainly we welcome your participation in this webinar and hope to see you engage and participate in the development of the knowledge base as well as the use of the information that's been placed there. And so with that, I'd just like to acknowledge lots of folks who are involved in AOP development effort within the EPA as well as many of our external partners, and then I'll turn it over and see if we have time for questions. Thank you.
Many thanks, Dan, for this very nice presentation. I will take the floor again for a very few concluding remarks. So, as you heard, we want to create more opportunities for researchers to become key players in this program. And researchers in countries are encouraged to support the development of the AOP and populate the AOP knowledge base with AOPs that will, in the future, support chemical risk assessment. A broader engagement and participation of scientific societies to the AOP development, review, and publication processes are key to the success of the program. In the coming months, likely in the second half of 2019, there will be another follow-up webinar dedicated to practical AOP development in the AOP knowledge base. And in the meantime, welcome and visit our website. Okay, so here are a few useful links to the OECD website. The adverse outcome pathways. This here you will find the future webinar announcements in addition to the recordings of the past webinars. So this webinar today will be available here on this website, and the online training material that also Kate presented earlier today is available here. These are the links related to the AOP knowledge base. And with this, I would like to thank you very much for your attention. And if you wish to contact the Secretariat, you can contact either Magda Sachana or myself, Nathalie Delrue. And with this, I would like to move to the questions. We have received a few questions. I will read them. We have received a few questions via the chat box. So the first question that I will ask to the speakers, is it possible and meaningful to integrate QSAR and chemoinformatics models with existing AOPs? Do the guideline, wiki, and tutorial have information related to this?
Hi, this is Dan. I can address that. It certainly is useful to link QSARs and chemoinformatic tools to AOPs. One of the things that we didn't touch on today, but the AOP wiki includes also or has stressor pages. And so specific stressors, whether those be chemicals or other types of non-chemical stressors, can be linked to specific AOPs in the wiki or to key events in the wiki that those chemicals are known to perturb. And that allows for essentially the linkage of the chemical agnostic or non-chemical specific information, the biological information captured in the AOP, to be linked to the chemical space and chemical domains that actually interact with the biological targets. And so through that connection, you can then link chemoinformatic tools or QSAR information into the AOP framework and identify chemicals that and the relevant hazards through that. And so there are tools that are building those connections. So, for example, EPA's chemistry dashboard links from chemical through bioactivity to relevant AOPs in the wiki. And we are trying to use more controlled vocabularies to describe the stressors in AOPs so that we can enhance the interoperability between those kind of chemoinformatic approaches and linking to the AOPs. So the stressor pages are described to a limited extent in the handbook, but that's one of the areas that is undergoing further development and hopefully will be increasing in utility in the near future.
Thank you very much, Dan. The next question, are there any standards for test or study quality for data entered into the AOP wiki? And is there any effort to double-check the supporting study quality?
I can, I guess, address that as well. Currently, there are no requirements to assess study quality when putting things in the AOP wiki. I mean, it's really on the users, or sorry, not the users, the developers to try to evaluate study quality as they're pulling together the information. Study quality is also considered to some extent in the peer review of the AOPs, but there is not a systematic effort in place for assessing the quality of all studies that may be cited as support for AOPs. And so that's one of the themes or ideas, principles behind the collaborative approach, the ability for anybody to see the AOPs, the supporting information, and to comment on that information, is that if others in the crowd know something about study quality, identify that a particular AOP may be citing poor quality support, they can leave comments about that. Those comments are publicly posted and are also available to the authors so that the authors can then go back and revisit. And so it really relies on the power of a crowd to help check those things. And so hopefully over time it evolves to the point where you get rid of any poor quality studies and really rely on the high quality support.
I'd like to elaborate on that a little bit as well. There's also while there's no specific criteria for which studies can be included as evidence, there are criteria for the weight of assessment calls. So it's unlikely that a single key event relationship will be given a weight of assessment call of having strong weight of assessment with one single poor quality study. It's usually an accumulation of evidence from various studies that would provide strong evidence to certain relationships. So while there's no specific criteria for single studies, it's definitely the kind of crowd strength, if you will, or accumulation of evidence or weight of evidence that really adds to the strength of any relationship in the knowledge base.
Thank you very much for your contribution. We'll take a few more questions before the end of this webinar. The next one would be, can you incorporate key events from different species models, for example, using a molecular initiating event and cellular responses in human cell lines with organism population effects in zebrafish?
Hi, this is Dan.
Yeah.
So, yes, you certainly can incorporate key events or initiating events from different organisms, and one of the aspects that goes into describing a key event and key event relationships is defining the domain of applicability, and that refers to the species or the taxa for which that key event is relevant, the sexes, the life stages. So all that goes into the description of the key event. So where those domains of applicability match up, or where the biology is sufficiently conserved between those different groups of organisms, that information from different species can certainly be used as supporting information for the AOP and basically coupled together.
And an excellent example of that was Dan's thyroid hormone pathway example that he had in his presentation. And another good example, not only is it cross-species comparisons available, but cross-tissue, cross-sex. Again, as long as the domain is sufficiently described, then yes, comparison across sexes, tissues, and species is possible. For example, the inflammation example that I showed you before was 3 AOPs that were linked together, even though they were across 3 tissues.
Thank you. So now, can the AOP concept be applied to diffuse MIEs, or is it best suited to only specific stressor receptor MIEs? And the commenter says, I'm thinking about endpoints such as skin or eye corrosion, where a substance may have a broad interaction on many molecules to lead to the adverse outcome for the organ in question.
I think AOPs can absolutely be applied to sort of these diffuse MIEs, and the examples of skin or eye corrosion, skin sensitization, different reactive mechanisms. There's many examples of molecular initiating events, or at least even if we don't know the exact nature of the chemical biological interaction, identifying associations between different chemicals and chemical properties and perturbation of biological functions. And so, those can be fairly broad groups, and there are a number of AOPs that define these rather systemic or diffuse sort of interactions. And that includes things like simple nonpolar narcosis, just bulk interaction of nonpolar chemicals with membranes, for example. There are AOPs that describe those interactions and the potential outcomes. So I think the framework can still be used to support that type of description as well. It doesn't have to be a very specific sort of molecule interaction with a defined protein and in a specific domain or a ligand binding situation or anything like that.
Thank you. And to conclude, maybe a very a simple one: can AOPs have branches?
I'll take a stab at that one. So this is a common question that comes up, that AOPs are often criticized as being an oversimplification of biology due to their linear constructs. And as Kate mentioned, the linear shape of an AOP is sort of an artificial limitation. It's just so that when with the wide, biology is complex, and if you wanted to say to develop an AOP that covers all of the possible branches of a biological pathway, you would never finish. So it was kind of an artificial limitation to say the development of an AOP is a single line, so that it has a clear beginning and an end. But yes, you're correct. Biology itself is not so linear, and so AOPs themselves are linear for the sake of making their development simplified. But when you combine them together, they become branch networks. So yes, an AOP can be branched, but if you think of a two-branched AOP as actually two linear AOPs being combined together to form a branch network, that's a way to look at it. So the linearity of AOPs is just for simplifying development, but basically a branched AOP is a small AOP network.
And just along those lines, there is actually some guidance in the user's handbook that describes at least the conventions for when branching is appropriate in terms of entering a new AOP in the wiki, and when we would prefer that they be represented as separate AOP pages. But there's definitely cases where branching is allowed and even encouraged.
Okay, thank you very much for this. I hope that you found this webinar useful and informative. I would like to thank you all for your participation. I would like especially to thank the three speakers today for their presentations and responding to questions. And to respond to a last question, if the presentation will be available, they will be available as the recording of this webinar, which will soon be available on the website. So with this, I would like to thank you all, and let's keep in touch. If you have questions, don't hesitate to contact me, and hope to see you in the next webinar on AOPs, and I hope that this webinar has stimulated interest, and that you will engage in populating the AOP knowledge base. Bye-bye. Thank you all.